So welcome to our webinar. This is Distance Learning for Cell Biology, a ready-to-use virtual laboratory for teaching mitosis. Here's a brief overview of our presentation today. I'm going to give you a brief introduction to the Allen Institute and where you can find our resources for educators. And then I'm going to hand you over to Graham, who's going to do an overview of some of the selected resources available at allencell.org that are especially relevant for educators. Then we're going to hand you over to Eric from Washington State, who has developed this virtual lab, a virtual lab on cell division using a publicly available image database. That's the allencell.org publicly available cell images. And then we have plenty of time for wrap up and questions. So in the footer of your Zoom window, you will notice that there are two places that you can type things in. You can type things in a chat, which is where we've been doing our introductions. We also have a place to type in Q&A. So if you have a question that you want to have answered by the panelists, please put it into the Q&A window. I'll be keeping an eye on that throughout the presentation. All right, and this is who's presenting today. So our first presenter is going to be Graham Johnson, the director of the Animated Cell Team at the Allen Institute for Cell Science. He came to the Allen Institute from the California Institute for Quantitative Biosciences at the University of California, San Francisco. And he received his PhD in biology in 2011 from the Scripps Research Institute in La Jolla, California, where he worked in Art Olson's molecular graphics lab, and he's also a certified medical illustrator. Our second presenter today is Eric Shelden. He's an associate professor in the School of Molecular Biosciences at Washington State and an academic editor for PLOS ONE. He received his PhD in zoology from the University of Massachusetts in 1992. He teaches freshman and senior cell biology courses, and his research is on eukaryotic cell stress using molecular and computational methods. And I am your moderator. My name is Caitlin. I am the training and outreach specialist at the Allen Institute. I work with our brain and cell science divisions. I got my PhD in neuroscience from the University of Washington, and at the Allen Institute, I work to train scientists to use our open resources for research and teachers like you to bring research experiences to their students using our open data. So a bit about the Allen Institute. We are a biological sciences nonprofit. We're located in Seattle. Our focus research areas are in neuroscience, the Allen Institute for Brain Science, cell biology, the Allen Institute for Cell Science, and immunology, our newest division. We also support cutting-edge research across the world with the Paul G. Allen Frontiers Group, which supports research, individual researchers and research groups. Across our institute, our scientific process is focused on tackling complex, broad, hard problems in the fundamental sciences. And our approach is guided by these three core principles, which I'll return to in a moment. And we generate data, knowledge, and analysis tools. We share all of our data publicly for anyone to use. To date, we've collected about 8 petabytes of data. That's 1.7 million DVDs worth that you can access on our website. And you can use that data for your own research to support the data that you've collected, and also to provide research experiences to your students using this data that we've made available. So as I said, we have these 3 core principles: team science, big science, and open science. Team science means we work on interdisciplinary teams. We have scientists from about 35, I think it is, different disciplines. We have big science, large-scale projects with robust, massive data. We spend a lot of time making sure that not only are we collecting great data, we're organizing on our website in a way that's useful to users. And open science. All of our resources are available online at brain-map.org and allencell.org. Today, we're going to be concentrating on allencell.org. So there were a couple of links in the email that you received that have the login information for this webinar, which we'll send out again afterwards. At alleninstitute.org/learn, you'll find some lesson plans from the Allen Institute and from other educators that use our open resources to help guide teachers and their students through virtual labs using our open data resources. We also have some other resources available there, such as posters that you can get for your classrooms when you're back on campus. At allencell.org/educational resources, you'll find links to some of the data resources, videos of a previous talk by educators, including Eric, on using our data in their classrooms, and some other resources there as well. A link to this featured lesson by Eric is available on both sites, and it was in the email that you received before you signed in. So today that we're going to be concentrating on ways to use our open data in your classroom. Most of our resources were developed with research applications in mind, with the notable exception of our visual guide to human cells, which you can see in this photo, which was designed for students. But they can be used in classrooms to provide students with these research experiences without the need for access to lab facilities that may not be available in all schools, and for distance learning. We recognize that high-speed internet is far from universal, but for many students, virtual experiments can provide a bridge to continuing their science education. And just as a reminder, the Q&A and chat are open. Please put questions in the Q&A at any time. I'll share any immediate requests for clarification with the presenters and save other questions for the end. You can also talk to each other using the chat. We also have some scientists from the Allen Institute for Cell Science who are in the chat. So please give them a shout out and they'll be available there as well. All right, so now I'm going to hand it over to Graham.
Thank you, Caitlin. Got my screen up, yes? Great, thank you. Today, I'll give you an overview of our Institute's scientific goals and the public resources that we provide. And I specifically plan to focus on the resources that pertain to education. Our website, allencell.org, which is the home of our scientific resources. On this site, we have a new page called Educational Resources. You can currently find this under the animated cell menu at the top of the website. The Educational Resources page consolidates links and it describes other pages that are on this website, allencell.org, that can be useful for teaching. The first thing on this page, after the overview section, you'll find a link to the most complete and ready to use resource that we have available, which is the CourseSource virtual lab that Eric is going to describe in detail later in this webinar. And further down on the page, we have videos from Eric and two other teachers, Tom Martinez and Carlos Goller, who came to the ASCB conference, which is the American Society for Cell Biology conference, to give presentations describing how they use web pages, data, and tools available on allencell.org in their own classrooms. So you can watch the videos on this page to find out more details about how they've integrated these tools. In the next section down on the Educational Resources page, we've provided links to the various pages on the website that those three teachers and many other teachers have told us that they use. So if you capture nothing else from my part of this webinar, just remember that there's an Educational Resources page on the Allen Institute for Cell Science scientific website, which is allencell.org, that will give you access to everything you're going to see and learn throughout the rest of the presentation today. This one page has all the links you'll hear about because it also includes a link to the broader resources that Caitlin described, which are on our parent website, alleninstitute.org, under this button here. And that link contains, for example, the free biology posters to hang in your classroom and another set of growing resources that will be useful for teaching neurobiology that stem from resources generated from the Allen Institute for Brain Science. In a few minutes, I want to show you what each one of these tools offers. And of course, for our main event, Eric is going to describe how to use the 3D cell viewer in the CourseSource virtual lab he developed with Erica Offerdell. But first, I want to give you a brief overview of the research that we conduct at the Institute to help you understand where this data comes from, why we gather it, and what will be available in the future. This part will take about 10 minutes and then Eric will take over. And one last thing, we're currently working on improving the usability of this entire website, allencell.org. The website has grown organically with us just adding things to it over the years, and it's become challenging for users to navigate because there's so much material there. So before I start the show, I wanted to ask you to participate in a survey that will help our user experience designer figure out how to best reorganize the site. And the link for the survey is currently at the bottom of the Educational Resources page. Let's get started. So I want to overview a major goal of the Institute, which boils down to integrating the structural anatomy of cells that we can see in our microscopes with other data to be able to infer and learn the physiology of cells. I want to quickly highlight some of the tools that we currently provide online, while also describing some of the more sophisticated tools we hope to make available in the future. And for most of our image data, we label only 3 structures at a time to minimize perturbation in the cells. And I'll go into great detail about what labeling means and what I mean by 1 at a time, etc. But for now, I just wanted to say that towards the end of the talk, I'll describe a variety of methods we've been working on to help integrate the independent structures together into more complete representations of whole cells, ultimately across scales of length and time, which can provide a much more useful teaching resource. For any new research, the first step is observation. To begin to understand how cells function, we first need to be able to see them and to describe all the components inside of them. At our institute, we dive deep into this first step of observation by building 3D atlases of whole cells. And what you just saw was volumetric data we collect on our 3D microscopes, which I'll describe in detail. And we've segmented out 2 cells from that data set. One of them is undergoing mitosis, and you can see the spindle microtubules, and the other cell is in interphase. So a lot of you watching this webinar can look at this image and tell me with a high degree of accuracy which 2 cells in this image are most likely to divide within the next couple of hours. But can anyone look at the other half dozen uncropped cells in this image and tell me when you think each of those cells is likely to divide or to do something else, like to migrate, differentiate, or even apoptose itself? What if I gave you the time lapse or let you watch 20 other time series movies of the cell same line under the same conditions for longer periods? Would you then be able to predict when each cell is likely to divide within an accuracy range of, let's say, 4 hours? Or if we layered on spatial gene expression data or other structures such as the centrosomes, instead of just seeing the microtubules and the DNA, would you be able to improve your accuracy? Could you predict any of the cells to within an hour? Or could a computer vision algorithm do any better than a human? So we believe that by observing and quantifying these behaviors across scales, we can find patterns to help us crack the code of cellular complexity. Ultimately, we want to work with the cell biology community to help connect all these predictions to mechanistic understandings as well. And that's really where the material that you're teaching in your classrooms can be connected to the type of data that we have online. So to address these types of questions rigorously at the Institute, we need a lot of reproducible data. And this slide provides an overview of our imaging pipeline that I'm just going to use as a reference map as we go through this. We begin with the cells, and our model system is the human-induced pluripotent stem cell, which has a variety of advantages you can see in the bullet list. These are the actual cells that Eric is going to describe later in his presentation. A major reason we use these cells is because they can differentiate into a number of different cell types for use in the lab. And at our institute, we currently study cells in both their undifferentiated states and as they differentiate into cardiomyocytes. When you do turn the cells into cardiomyocytes, they eventually beat beautifully in a dish. And this top image is just showing footage shot through an iPhone through one of the dissecting scopes in the gene editing lab of the cells, tearing themselves off the substrate and beating in synchrony. And because we tag the components inside the cells, you can see the high resolution detail images at the bottom where GFP labels provide the details of the structures in the living tissue. To visualize our cells in a semi-automated manner, we grow them on 96 well plates where the individual cells settle and begin to grow and divide into colonies that look like this at low magnification. And here we're just using an edge detection algorithm to identify and color code the different colonies with their boundaries. But if we zoom into a colony, we of course find that the cells are practically transparent, and our HIPS cells look like this at higher magnification and white light. To make a component pop out from the background, we use fluorescence microscopy on cell lines that have structures which we have endogenously tagged with fluorescent proteins. To give you a very quick review of endogenous tagging, We start with a wild type cell line. So on the left-hand side of your screen, I've got a cross section of one of the cells. You can picture it segmented out from the colony. It's got neighboring cells attached to it and a matrigel substrate on the bottom. There's a very simple nucleus illustration, mitochondria as the organelle that's going to be targeted, and an overly simplified representation of DNA on the left. So all the native proteins are generated through normal processes of transcription and translation, because we have the normal number of chromosomes in our cells. The proteins localize to their appropriate compartment through a variety of mechanisms, of course, depending on which compartment they end up in. But when we look at the cells under a microscope, we rarely get the resolution and contrast we need to be able to distinguish structures from the background or from each other in a useful way. So this is the wild type cells that we start with. But we can use a system like CRISPR-Cas9 to insert the gene for a fluorescent protein directly into the amino acid sequence of the protein that we want to tag, which results in a protein expressed at endogenous levels that has GFP attached to it, a little green glowing guy off to the left. So when the protein ends up in its native location, we can use fluorescence microscopy to make that location stand out from the background, which looks like this, or we can merge that fluorescence image with the bright field image to see the relationship between the two modes and between the mitochondria and all of the other structures. So this gives us beautiful data, which looks like this for a cell line where the nuclear lamina was made visible by tagging lamin B protein. That's the inner shell lining the interior of the nuclear envelope. We collect the data in 3 dimensions, and we have a growing quiver of microscopes at the Institute. But our initial and primary workhorse is just the spinning disk confocal microscope. And these are examples of Z-stack movies, along with a couple of time series movies in the bottom, showing data for 18 of the different cell lines that we have available. These are all the cell lines that could fit in one screen, but we actually have 30 lines available on our cell catalog, which is available on allencell.org. And there's also a very useful resource where you can find these types of Z-stack movies as well as time series movies of each one of the cell lines in that cell catalog, which has the URL at the bottom. So a number of organizations around the world have started using lines from the cell catalog to study systems in basic cell biology like we do at the Institute, but also developmental biology and in various human diseases. And that type of connection to human impact can often be motivational for a lot of students in the classroom. So in the beginning, I told you I would describe approaches we're taking to integrate images collected from independent cell lines. So on the left-hand side of your screen, you see the names of some of the proteins that we have tagged. These proteins were selected to be the minimally disruptive proteins that go to specific organelles within the cell to highlight those organelles, which is appropriate to the resolution of the spinning disk confocal microscopy. And in each cell line, by default, we dye the DNA, we dye the cell membrane, and we endogenously tag a single protein one at a time. So how do we then take all those individual images of proteins and put them back together to create, for example, the cell in the middle with 15 channels all visible at once? So our first step towards that is our automated microscopes collect tens of thousands of cell image fields. And I'm showing a sample here of the cells that have been segmented from those fields. And the first step is to just provide access to the data for all those cells. And this is very much introduction to some of the tools that Eric is going to describe in a few minutes. We provide both online tools and desktop tools for visualization of volumetric data. And I'm just showing a couple of them here, and we'll zoom into one of them, which is called the Cell Feature Explorer. Again, the links to this are provided on that website under educational resources at allencell.org. And this shows you how in a website, without downloading any additional software, you or your students can look at measurements taken upon the cells. And as you roll over each one of the dots, each dot is an actual cell. You'll see a max intensity projection image of that cell. You can click on the cell to start to build up a gallery on the right-hand side. In this case, I'm choosing measurements of nuclear volume versus cellular volume, where the cluster of mitotic cells tends to fall out towards the upper left from the interphase cells in the lower right. And when I click on one of the cells that I've selected from that collection of 32,000 cells in the gallery, it automatically loads into a volumetric viewer right there in your web browser. And all of the basic features you would expect of a volumetric viewer are available to you, such as adjusting the transfer functions to make the brighter pixels stand out more from the background noise. So that was the Cell Feature Explorer, and Eric is going to give you, or he's going to describe how he uses an older tool that we developed, which is called the 3D Cell Viewer. For this, we inherited an open source code from UC Santa Barbara. It uses the same approach to show volumetric data directly in your web browser, and it's a cell field centric gallery, whereas the Cell Feature Explorer is focused on one cell at a time, and then you can move up to see the entire field of cells that single cell came from. But this one is focused more on the whole cell fields. So it again has the, whoops, sorry. I'll just move on past that because Eric's going to show it. I can't get my movie to play. But in the beginning, so another method of integrating structures aside from just presenting all the data and making it easily available is some projects we've actually worked on at the Institute. And one of these is called the Integrated Mitotic Stem Cell. So at imsc.allencell.org, you'll find what we call a scrolly-telling page, where you can click through about a 20-minute description of how data is collected at the Institute and how we have taken these individual cell images and integrated them into a 15-channel cell that has 15 different organelle systems described over time as they change throughout mitosis, and some key observations that we've made from that process. So again, that link is available on the site. And we have an earlier tool that we've had available since 2018 that Caitlin described, where we want to show the most complete cell possible using all the technologies we had available at the time. And this is primarily using a technique called label-free determination. This is a deep learning process where having tens of thousands of microscope images available, we're able to actually predict relatively reliably what the structures of several different organelle systems are directly out of the white light bright field image. And that allowed us to construct a 14-channel model where 3 of the channels are from observed data, including the DNA, the cell surface, and the microtubules. Again, this might be confusing looking because these are microtubules at the resolution of spinning disk 3D microscopy, not at the structural resolution that you might be used to teaching with. I'm going to fast forward this a bit. But what we have is when you dig deeper into any one of these structures, not only can you see all the details of what it looks like as it undergoes mitosis, on the right-hand side, there's a description of the organelle function, the organelle morphology, and illustrations that can relate it back to the more structural perspective of that morphology. So that again is linked to it, and we're gonna, at the end after Eric presents, we will summarize where all these tools can be found. And now, without further ado, I'd like to introduce Eric.
Good morning, everyone. It's a real pleasure to be here. I hope everyone is safe and healthy, and all your families are doing well. These are interesting times to be doing science and education. So the lesson that I'm going to discuss has been published in a journal called CourseSource, and that's linked, that you have the link to that. And there's also a link on the Allen Institute's website to a seminar I gave about this. And so you have access to all of that. And I'm going to just give you kind of a brief overview and share some of the insights that we've had over the last couple of years using this exercise in a classroom. And so I'm gonna, without further ado, see if I can get you over to my presentation. Yeah, there it is. So as you've heard, the Allen Institute has just a tremendous array of really powerful and intriguing data and data visualization tools that are available to everyone, you and your students and researchers across the world. And I've been teaching a senior level cell biology course online as a distance learning class for a number of years. And what I wanted to do was to develop an exercise that was reminiscent of an undergraduate biology lab classroom activity that used this resource and also gave some insight into real science. And this is what we kind of came up with, which is the virtual lab on cell division. And I should say this was a collaboration between myself and then Dr. Erica Offerdahl, who's associate professor at Washington State University, and her research is STEM education and STEM pedagogy, and then Graham, who you've been hearing from. This is an overview of the laboratory activity and its learning goals. So we first asked students to do what they do normally in a cell biology class, which is read about cell division. And then we asked them to think a little bit about the image data that they're going to be presented with and how it was obtained, and write and justify a prediction regarding how many cells they're going to see at each particular stage of cell division based on what they understand cells are trying to accomplish at each one of the stages of cell division. So prophase, for example, the cell has to take apart its interphase microtubule array and disassemble the nuclear envelope and so on and so forth. And what we're trying to do is get them to think about cells as living things that have to perform a functional activity rather than just a series of terms that they have to memorize. And then we assign them the task of actually going about doing this analysis. And this is very reminiscent of what is actually being done in my laboratory, where, for example, we're interested in how different perturbations might affect the cell's ability to go through mitosis. So aside from the collection of the images at the microscope level, what undergraduates and graduate students in my laboratory are doing is actually this very same thing. And then we ask students to analyze their results and then to discuss the results and with respect to their predictions to see whether the predictions were true or not, and if not, why not. And so our goals initially is, of course, just to have students understand the process of cell division and sort of have a deeper connection to mitosis than they would gain by just reading a series of steps in a textbook. But then more advanced students, we might ask them to think a little bit about how biological data is collected and what sampling methods, strengths and weaknesses of sampling methods, and even some statistics. For example, if students are assigned a relatively small number of images to analyze, then the results they're going to get are going to be very different from each other. And if we combine the data from several students, they might get a more accurate picture of what's going on in the data set. And then finally, to get some real experience working with actual cells. And surprisingly, even seniors may have gone through 4 years of biology education and never seen an image of a real cell. And so sometimes they're actually kind of initially a little bewildered by what they're looking at. So that's kind of a neat experience for them. And we wanted to make this relatively scalable. So we've run this exercise in a senior level distance education course, which has about 20 students in it. every semester, but more recently this semester, with the distance learning necessitated by the current health crisis, this was assigned to an undergraduate 100 level biology course that had over 500 students in it, and it worked, I think, pretty well. So the implementation is the thing that we needed to be able to do was to be able to assign students images to go analyze, because otherwise there's just an awful lot of stuff on the Allen Institute's website for them to explore kind of endlessly. And so the way we implemented this was to links that encoded specific images that they could then go to and analyze. And that's by virtue of this URL, which you see down here. And this, this link encodes basically this screen, which you see here. And we set this up as a spreadsheet. This can be an Excel spreadsheet, and it turns out it works pretty well in Google Sheets as well. And so what the student gets is a spreadsheet like this. And and to make this,
hey, Eric, we lost your audio for about 10 seconds there. If you could just back up real quick.
oh, sorry, thanks. Yeah, so this is images of the spreadsheet that students receive. The links are hidden, and you can just click on each one of these terms, image one, and so on and so forth. And that will cause your web browser to open the image directly so that you can analyze it. And some other resources that we developed to make this easy for instructors to implement are available on the CourseSource website. We have an editable handout with some instructions on using the 3D cell viewer that is in a Word doc format so that you can modify as you wish. There's a PowerPoint mini lecture on cell division that goes through the various stages of mitosis and tells you a little bit about what is going on in each one of these stages. There's another PowerPoint presentation that has a fairly large number of example images that have been pulled out of the data set to illustrate what each cell stage looks like in the data set. This is one thing that students often take advantage of because for the most part, one of the difficulties students have is confusing interphase and prophase nuclei. And you can't really see that here, but prophase, of course, the chromosomes condense, but in interphase, the cells have quite a bit of texture in their nuclei as well because of the distribution of euchromatin and heterochromatin. And students are kind of taught in textbooks that interphase nuclei look featureless, that they're just this round ball, and actually real cells don't look like that. And then we have some Excel spreadsheets. There's an example student file that you can copy and paste into an email and send to students. There's a link to an Excel spreadsheet that's got links to all possible tubulin stained images. So these all are images that have chromatin in blue, and then the mitotic spindle because of its labeling of the microtubules in the cell. So there's 927 possible multi-cell fields that you could potentially assign to students. And all of them are linked in this spreadsheet. And then there's a third spreadsheet where we've actually gone through and tallied how many cells are at each stage of mitosis in the field of view. And I should say that when I teach this, I don't really emphasize that students have to come up with the right number of cells at a particular stage. I'm much more interested in their thought process and that they sort of go through the exercise. Okay, and then the last thing is I like to assign every student their own set of images. And if you have 60 students, this can be kind of laborious to come up with these Excel spreadsheets. So I wrote a Java application that's available on SourceForge. And what it does is it takes a text list of students' names. You indicate how many images you'd like to assign each student, and then it generates a personalized list of these Excel spreadsheets with links to the Allen Institute's images. So just some nuts and bolts about the exercise. Learning time, I generally assign this to take about 2 hours. I ask them to spend about a half an hour, or I suggest they spend about a half hour thinking about the cell division. They've presumably been taught this ahead of time, and then writing a hypothesis statement, and I grade that hypothesis statement. And then I figure it takes them about an hour to count 20 or 30 cells, images worth of cells, and then write a summary. And some students take more than 2 hours, but others finish in less time. For a grading rubric, what I'm looking for is a well-written, logical, defendable hypothesis. I don't necessarily care that it's correct. And an example with that would be a statement that says prophase is going to be the longest stage of cell division because the cell has to condense its chromosomes, assemble the mitotic spindle, and disassemble the interphase microtubule network. That shows a lot of insight, a lot of thought about what's going on in the cell. If sometimes you get students that say, I think that the order of the cell stage is going to be prophase, metaphase, anaphase, telophase, and you'd like them to know why. And some of them write something that comes from the textbook and doesn't actually illustrate that they've thought about it, and that would I give them low marks for that. And then the data analysis, if they produce charts or graphs or some sort of summary that shows insight into the data, I give them a lot of credit for that. Sometimes they just assemble, sometimes they graph the wrong things, or they don't compile the data in a meaningful way, and I give them less credit for that. These are some images, some examples of what students have produced. Most of them look very much like the kinds of illustrations that you'll see in textbooks, and I think it really gives students a sense of empowerment.
Eric, we've lost your audio again.
Okay.
Yeah, just back up about 20 seconds.
Okay, so here are some examples of things that students have produced, and they look a great deal like the illustrations that you might find in a textbook. And I think it's quite empowering for students to produce this on their own, from their own analysis, and see where these kinds of illustrations come from. Obviously, a lot of students find that the majority of cells are in interphase, and that's what you would expect. Most of them do find that prophase is the most prevalent other stage, and I still think that that's probably due to the confusion of looking at cells that are real instead of just illustrations. Since when I do this, I don't necessarily see that. The next most prevalent stage that students find in these data images is metaphase. And this always surprises the students, because when we teach metaphase, we tell them that it's basically just the cells line up the chromosomes at the center of the cell. And so we kind of imply that not much is going on, but it's also the site of a really important cell cycle checkpoint. And the cell has to pause there to make sure all of its chromosomes are where they're supposed to be. And this takes quite a while, and that actually shows up in their data. So it's kind of an inspirational moment for the students when they realize this from their data, and they get kind of a kick out of that. Then conclusions, again, I kind of like them to illustrate some thinking about this. Many times the students' expectations are not met, and I like to see them think their way through why.
Oh, we've lost you one more time. So if you just back up to the students who get 0 points.
Right. So as I said, my emphasis in all of this is really students illustrating a logical thought process in analyzing their data and aligning their conclusions with the data that they produced. And then here's some examples of student feedback, and I think they illustrate that for the most part, students really enjoy this exercise. A lot of times they have never really seen real images of real cells before. The cells are human cells, and so that gives students a lot of personal connection very often to the material. The kinds of comments I get are, that it was beneficial to look at raw data. Often that the exercise makes the sort of abstract terminology much more accessible to them. They like the insight into cell biology, methodology, and data collection. And I think most important is that it really gives a sense of reality to the students' learning of cell biology. So I'm just going to really briefly finish up by showing you an example of how the spreadsheet works. This is the Google Sheets document that you've been provided a link to. And all you're going to do is come over here and click on these things, and that should open up in the web browser the specific image that students are supposed to look at and analyze. The instruction document gives some ideas about how students can go about counting the various stages on different platforms, but you can turn on and off the microtubule stage. This is obviously a prophase cell, and here are these interface cells that show a lot of texture. And I've got this image as well. This is obviously a clear example of a metaphase cell. So students can kind of explore these images and analyze various components of the cell. So I'm going to turn this back over to Graham to finish up before we ask any questions. But I'll be happy to answer questions afterwards.
Great, thank you, Eric. Quick confirmation on my screen. Okay, great, thanks. So we're about 4 minutes over, and I'm just going to give you a brief summary to show you where the links are again, and then we'll go into a question and answer session up until the end of the hour. So today, I hope you learned from Eric that students can use open online databases to conduct experiments virtually. And while virtual experiments can't replace live experiments, they can introduce new types of data or serve as a bridge for distance learning, which might be especially useful given the conditions we're all in right now. As a reminder, everything we presented here is on the site at allencell.org/educational resources. The tools were first developed with researchers in mind, but when we see how they've been adopted by people like our three speakers, the three teachers who are giving the presentations on this website and Eric today, we can clearly see the potential of this material for education. We have some tools that have broader language, but we've not yet developed any tools specifically for educational audiences. You can find links to the resources on allencell.org. or on alleninstitute.org, as described earlier. And we're now going to switch to a question and answer session. And Caitlin will read some of the questions that you guys have presented, and the three of us will answer any other incoming questions that we see. But as a reminder, we're going to move all the Q&A as well as the questions that have been answered, as well as any remaining questions we couldn't get to, over to the page forum.allencell.org later this afternoon. And I want to thank all of the people at the Institute who do all the hard work, and Paul Allen for his vision in creating an amazing institute to do important science. I want to thank Eric for his time today, Caitlin for putting this together, Allison and the IT and the rest of the communications team as well for helping us get this up and running on Zoom. I'm going to stop sharing and switch back to Caitlin now.
All right. So thank you to everybody who has been putting questions into the Q&A. We're going to get to as many of these as we can, and the rest of them we will answer in the forum. There will be a link to the forum in the email that you'll receive after this webinar, or you can go to forum.allencell.org. So our first question to answer live is, Eric, do you distinguish between mitosis and cell division cytokinesis? For example, liver cells are often binucleated. Then a question for Graham within that, does the Allen Institute have liver cells and other oddball tissues?
So to answer that, in my coursework, I do distinguish between cell division, mitosis, and cytokinesis. And actually, we use the Drosophila embryo and epidermis as an example of cell division without. Cytokinesis doesn't show up in these images, so it's not one of the stages that cells can look at.
And I'll chime in. Where did that question go?
In the answer panel.
Okay, so at the Institute, we've decided only to focus on one differentiation protocol, and that's to cardiomyocytes. And we want this to be a template where we will provide the methods for other labs to do their own differentiation into other tissues. We do not have liver, but we do have the cardiomyocyte data coming off the pipeline now, and we're working on ways to analyze it and then to provide it to the community the way that we do provide the human-induced pluripotent stem cells in their colony state.
Great. Our next question comes from Rich. Are there virtual microscopes that a student can use?
Not quite sure what you mean by virtual microscope, but if you mean a microscope that a student can control, we don't have that. I think the short answer is no, not on our website. I think there are other websites that offer that. We have considered having streaming footage from our microscopes that you could watch on a live cam, and if that's something that would be interesting, we can provide it. But in terms of being able to control a microscope remotely, we don't provide that. Or in terms of having a virtual microscope where you're zooming into an artificial tissue, we do have a project called Simularium in development, which is going to be a multi-scale viewer that will take you all the way from tissues down to mechanical simulations of molecular interactions, so you can understand how emergent complexity produces complex behavior in cells. But it'll be a couple of years before that's going to be useful to your students at the level I just described. We'll have some early previews of it coming up in a few months. Thanks.
And then the next question is from Brett. Does the Cell Explorer or other databases at the Allen Institute include ultrastructural details such as TEM or immuno-EM? I will chime in and say that the Allen Institute for Brain Science does have an EM project. They have not released that data yet, but they do work on that. I can share in the forum a link to a story about that.
And on the cell science side, the Cell Feature Explorer doesn't have a connection like that, but some of the other tools, such as the Visual Guide to Human Cells, includes some classical electron microscopy imagery that comes from the textbook world of what these structures look like at that resolution, as well as illustrations from the protein data bank of what the protein structure and the macromolecular structure. So we're talking alpha tubulin, beta tubulin dimers and how they relate to whole microtubules, for example. Again, we're looking to start filling in this multi-scale view of whole cells with the Simularium project, where we want to integrate image data with modeling data into a comprehensive virtual cell And again, that's going to be a long-term project, but keep your eyes open for some previews of it later this year. Thanks.
The next question is from Luria. Does the cell viewer work on a tablet or iPad? This question was already partially answered by Dan in the chat window, who said that the Cell Feature Explorer and 3D Cell Viewer are capable of running on tablets as they are ordinary websites, but are not specifically designed for small screens or touch interaction. Graham, do you have anything to add to that? And Eric, do you have any comments from your students who might have been using touch devices to complete this assignment?
I'll quickly say that the 3D Cell Viewer that Eric uses in his demo functions. It's not an optimal experience because the viewer for the window where you would do the counting is relatively small. The Cell Feature Explorer also functions, but the plotting interface of the graph is also relatively small on the screen, so it's not very useful yet. This is a priority for us to develop later in the year, but I'm really curious to hear if Eric has experienced students trying to use it and had any success, or if they've always had to go to their laptop.
I can actually not share any of that. Not aware of.
Did you say that you are not aware of students who have used?
Yeah, students have typically not had technical problems, but I cannot tell you what they're using to do it.
Great.
We would love to hear in the forum if that is a major priority. For example, what percentages of students do you have in your classrooms that are using those types of devices? Thanks.
I think we have time for about 2 more questions. And again, any questions that you post in the Q&A window before the end of the webinar that we don't get to, we will answer them in the forum. So you will get those answered. Our next question is from Rich. Is it possible to see mono-oriented chromosomes?
Eric, if you can tell me what a mono-oriented chromosome is, I can answer that question.
Not sure, to be honest with you.
If I had to guess, sorry, I can't look this up. We can answer it with our scientists on the forum later. You might be talking about the karyotyping images that are available on the as part of the quality control measures we take on the cells that are available in the cell catalog. And otherwise, I'll try to follow up with once you can clarify that question or when I can look up the term. Sorry about that.
Rich, if you can just pop a clarification to that question in the chat, or once we post it on the forum, post a clarification there, we will get you the answer.
I'm going to say that the answer is probably no. And because the reason I say that is because the resolution of the chromatin images is certainly adequate to tell what stage the cell is in mitosis, but it's generally not good enough to see specific chromosomes.
Great, thank you. So our next question is from Jane. Does the Allen Institute ever use Zooniverse or other citizen science online platforms to crowdsource interpretations of cell observations over many cells or tissue types to help inform scientific research at the Institute? So I will hand this over to Graham if there's anything for cell science, but I will say that the Allen Institute for Brain Science, some of their 3D neuron reconstruction data is available through a citizen science platform called Mosaic that enables people to trace the neurons in 3D, and that data does get fed back to the Institute. We can post a link to that in the forum. That's a brain science resource. It's using a different data set than what we were covering today, but may still be of relevance to your classes.
This is a very timely question because we just have started talking to the developers of Zooniverse, and we are testing how we can use it not only for citizen science, but even as a powerful tool at the Institute for doing some of the image quality and image sorting and annotations in an efficient manner for our scientists. And then we want to eventually try to compare that to citizen science efforts. So we're very excited about that. And we'll be sending out, I'm sorry, I'll leave it at that for now.
Great. And our last question that we will answer live, again, if you have more questions, please pop them into the Q&A and we will answer them on the forum. is I love the available data and the mitosis lesson plan. This is from E. Batchelder. What other lesson plans are you working on, or any ideas that you've had that we might be able to run with on our own? So at alleninstitute.org/learn, we have a couple of additional lesson plans besides Eric's. We have a couple that were developed at the Allen Institute that I worked with some of our scientists, and we have those available. And we have a couple that were developed outside of the Institute. We have one brain science one and two cell science ones that are all available for you to use. If you do end up using the resources in your classroom and you write up a lesson plan that you've shared publicly, whether it's in CourseSource or another forum, please shoot us an email at communications@alleninstitute.org to let us know.
And I want to add to that at the Institute for Cell Science, none of us are teachers. We're biologists and engineers. And we rely on teachers like Eric and Carlos and Tom to come to us to tell us how they're using these tools. We are very interested in ways that we can modify or develop these tools or test them for educational uses. And if there are successful uses out there like the one Eric demonstrated today, we want to help amplify that, as Caitlin mentioned as well, and get it out to as many people as possible. So we're really interested in continuing this discussion in the forum and hearing how you guys are using all this. Thank you.
We are just about out of time. Again, you can post some last couple of questions in the Q&A. We'll transfer those over, or we'd love to see you over in the forum to continue this conversation. Everything that we've talked about today doesn't have to end now. We would love to hear about how this comes to fruition in your classrooms. Please keep your eye out for an email from us in the next day or so with links to all of the resources that we've talked about today, a link to the forum, a link to the video of this webinar, and more. Thank you all for tuning in, and I hope we see you at a future event or virtual event from the Allen Institute.